Good morning, everyone. Neljean from Team Red BMLS 2A. Our topic for today is all about Citrobacteriaceae. Okay. So, Citrobacter came from the Greek word citrus, meaning lemon, and the bacter, meaning a rod, or a citrate utilizing rod. Natawag siyang citrate kasi all species of Citrobacter mag-grow siya sa Simon citrate medium. So, the general characteristics of the Citrobacter, they are gram-negative rods in single, pairs, short chains, aerobic or facultative anaerobes, non-endospore forming Enterobacteriaceae. They are oxidase negative and catalase positive. Late lactose fermenter or ginatawag na sila paracolon bacilli or colored colonies at reduces nitrates to nitrites. So we have 11 species. First, we have Citrobacter friondi, Citrobacteriaceae, also known as Citrobacter diversus, Citrobacter amalonaticus, Citrobacter youngii, Citrobacter bracchi, Citrobacter farmeri, Citrobacter gelani, Citrobacter mordiniae, Citrobacter rodentium, Citrobacter silaki, and the Citrobacter workmanii. Okay, so mag-focus lang ta sa Citrobacter friondi, Citrobacteriaceae, also known as Citrobacter diversus, and the Citrobacter amalonaticus. Okay, so next, pathogenesis. The pathogenicity of each enteric member may be determined by its ability to metabolize lactose. So first, we have Citrobacter friondi. So Citrobacter friondi naa siya duha ka antigenic structures. First, na O-lipopolysaccharide antigen 29 by viral lens capsular antigen. For O-lipopolysaccharide antigen 29, cross reactions occur. This can be due to the lysogenization with phages or because of the shared epitomes on the lipopolysaccharide expressed by organisms such as Salmonella and Citrobacter. So the O antigen can be detected by boiling or autoclaving the strains to overcome the inagglutinability caused by K antigen. For the bicapsular antigen, it was first described by Felix and Pitt. So the production of bicapsular antigen is chromosomally mediated in this bacteria. The bivirulence antigen is a surface polysaccharide which is formed by most of the strains of Citrobacter species. Okay, when fully developed, it renders the bacteria agglutinable by Vi antibody as well as inagglutinable by O antibody. The Vi antigen is heat labile and destroyed at 100 degrees Celsius within 60 minutes. Also, the mutants deficient in Vi antigen arise spontaneously and convert the strain into Vi lucky. These mutants are called W forms or typhi forms, and the variation as V2W. So, ato ang Citrobacter rodentium and some strains of Citrobacter friondi na sila EAA gene. EAE or enteropathogenic E. coli attaching and effacing gene. This gene is for entenum, which is required for the formation of the attaching and effacing lesions by Citrobacter rodentium. For Citrobacteriaceae, expression of 32 kilodalton outer membrane protein was found among isolates of Citrobacteriaceae. So Citrobacteriaceae is easily engulfed by human macrophages but survives phagolysosome fusion and replicates within them. Okay. Next. So these species rarely cause primary infection in healthy individuals, but the immunocompromised patients are at risk for nosocomial infections. So the Citrobacter species are considered inhabitants of the GI tract, other animals, and may cause opportunistic infections. So the Citrobacter friondi, also known as Balaropvetesta group, can be isolated in diarrheal stool cultures. Although it is a known extraintestinal pathogen, its pathogenic role in intestinal disease is not established. So Citrobacter friondi has been associated with infectious diseases acquired in hospital settings such as UTI, pneumonia, sepsis, intra-abdominal abscess, and endocarditis in intravenous drugs abusers. So recently, an outbreak of gastroenteritis and hemolytic uremic syndrome was associated with Citrobacter friondi producing a verotoxin 2, also called Shiga toxin 2. So, kada siya, ng picture dyan sa kilid, muna siya ang example of gastroenteritis. And sa ubos, ang hemolytic uremic syndrome. Okay. So for Citrobacter casei, formerly known as Citrobacter diversus, is a pathogen documented as the cause of nursery outbreaks of neonatal meningitis less than 1 month of age, as well as brain abscess. So the transmission of Citrobacter casei could be from mother to fetus. Okay. Lastly, we have the Citrobacter amalonaticus. So it has been isolated from the sites of extraintestinal infections present in blood and wounds. So Citrobacter amalonaticus frequently found in the feces, but no evidence has been found that it is a causative agent of diarrhea. Okay. For the laboratory diagnosis, they're gram-negative rods arranged in single, in pairs, or in short chains. They ferment lactose, producing colonies on mock agar, and a catalase positive. So, kanamo na siyang gram-negative rods, color pink, or red, pink to red, something ano. And catalase positive. Okay. Or the colony morphology in BAP or blood agar plate, they're gray color, smooth, and moist. So, nakipicture mo na siya. And for McConkey agar, lactose fermenting bacteria, pink colonies. Ayun. The colony morphology in eosine methylene blue agar, ano siya, greenish metallic sheen. Next, in Salmonella shigella agar or deoxycholate citrate agar. So, muna siya ang Salmonella. Ang colony morphology nila kay black-centered colonies with clear periphery. For the TSI agar, na siya AA pattern with positive gas and hydrogen sulfide production. Other tests are urease, which is positive or negative, mannitol positive, lysine decarboxylase negative, indole negative, methyl red positive, VP negative, citrate positive, and the motility is positive. Outline of biochemical tests for separating selected enteric genera. So the colonies from selective media calculated into triple sugar iron or TSI, rapid lactose fermentation on TSI. For the positive in lactose, we have the Erwinia, Citrobacter, Enterobacter, Klebsiella, Willianella, Escherichia, and Klevera. So positive for the motility, we have Erwinia, Citrobacter, Enterobacter, Escherichia, and Klevera. Positive in indole, That is Citrobacteriaceae clevera. And for the negative, you also have Citrobacter species, Erwinia, and Enterobacter. So hydrogen sulfide can be positive or negative ang Citrobacter, and citrate positive as well as LDC negative. So that's all for today. Thank you for listening. Bye.